Okay, I ended last time talking about the root system in plants, and this time I want to switch our attention to the above ground portions of the plant and the shoot system. So just as a reminder, remember that the shoots is includes the above ground portions of the plant. That includes the nodes where leaves and branches are attached, and the inner nodes in between those nodes, and their series of apical buds, and then axillary buds where that growth is occurring. As well as, of course, the photosynthetic organs like the leaves extending from that plant stem, that plant body. And so really, when we think about the shoot system, this is a repeating series of nodes and inner nodes. Nodes, inner nodes, leaves, and apical and axillary buds. And importantly, as a plant grows, it does this by adding these parts, so increasing the number of these parts. Increasing the number of these parts. Once a leaf or a node or inner node forms, it really doesn't get any bigger. It is what it is. Remember that our growth is happening our growth in terms of not adding new cells is happening in that apical meristem where cells are dividing. So these are growing from the tips. And once the parts, flower parts, leaf parts, branches are formed, they really don't get much larger over time. It's really about increasing the number of those parts. And so rather than increasing the size of those parts. So plants grow by adding more nodes, inner nodes, leaves, etc., not by increasing the size of them. The plant grows by increasing the number of these parts, not size. And of course, there's a huge diversity. There's a huge diversity in the shoot system. And so here I've got two of our records. And what I mean by our records is the records in terms of the size of the shoot system. And so on the left-hand side here, we have the smallest plant, the smallest plant. And this is duckweed. Many of you probably recognize this from experiments that are done in our 114 class. Duckweed's in the genus Lemna. And this is the shoot system right here. This little tiny stem with two leaves coming off of it. That's our shoot system. We actually can see part of the root system down here. And so this has an extremely small, very tiny. This is the smallest flowering plant is this duckweed. On the right-hand side, we actually have the largest organism on Earth, and this is in the giant sequoia. So in case you can't see that, here's a person, that's an adult-sized person there at the base of this tree. These are incredibly tall, incredibly big. And so this is actually the largest organism on Earth in terms of biomass in these giant sequoias that grow in the mountains of California. And so you've got really huge diversity ranging from these teeny tiny little plants to these ginormous, huge plants of the giant sequoias. Okay, and so the way that branches are actually added to the way that branches are added contributes to that overall shape and size of the shoot system, of the above ground portion of the plant. And so I just want to show you this in simple example of how branching angles affects the overall shape of the plant and why. And so we have an example on the left here of very short internodes and wide branching angles. And wide branching angles. And on the left, on the right-hand side here, we've got the opposite of that. We've got long internodes. And narrow branching angles. And so what this does is actually changes the way that these species are going to be harvesting light. And so in varying different environments, it might be better to be a shorter overall plant with wider branching angles, or something where you're a taller overall plant with longer internodes with these narrower branching angles, depending on what other plants you're interacting with and how the situations of the particular environment. And so this variation is really important because it allows, so this diversity or variation allows for different species of plants to harvest light in different ways. Variation allows different species to harvest light in different ways. So harvest light differently. And why would this be important? Well, this is important because it will then minimize competition if these are growing together. When these are growing together, this reduces the amount of competition and allows different plants to thrive in many different environments. And so I want to show you one of my favorite examples. I alluded to this on the sort of title slide of this section, and that is in the Hawaiian silver swords. This is a group that the whole monophyletic group are called the silver swords. So this is a monophyletic lineage. That has an incredible diversity of growth forms, ranging from trees, like we see in this Debaudia linearis, to small cushion plants, like we see in Debaudia wailelile right here. And so we've got these two things, and two plants in the same genus that range from large trees to small cushion plants to these little herbaceous plants to sort of edge species and shrubs. And so all of these are part of the same genus, but have a huge diversity in their shoot systems. And so it turns out that in the Hawaiian silversword group, we have the closest relatives of these are actually a North American species called, which is in a group with the common names of tarweeds. And so these tarweeds are these small little annuals, so meaning that they go from seed to seed in 1 year. And so this small annual herbaceous plant is the closest living relative to the monophyletic group on the Hawaiian Islands called the silver swords. And so presumably 5 million years ago or so, or a little longer than 5 million years ago, an ancestor of these tarweeds, something similar to the tarweeds, dispersed to Hawaii, one of the older islands that's now submerged. And then being in a new place with lots of different open niches to exploit, these actually speciated into all of the available habitats and available niches. And during that speciation, or that radiation of species, you actually end up adapting to very specific habitats, and the shoot systems kind of mirror the specific environmental requirements or environmental pressures of each of those habitats. And so in some places, like out on these barren lava flows where the cracks are the main place where moisture and soils accumulate, we end up with species that are small herbaceous plants that live in those cracks. In the wetter forests, we end up with trees and or smaller or shrubs. In the exposed high elevation areas, we end up with these rosette plants like the silver sword. That's what this whole group is named after, these incredible plants, the silver swords themselves, and or these small cushion plants. And so these are all having these different chute systems to be able to, that are adapted to specific environmental, specific ecological conditions. And so this is something that's called an adaptive radiation. And silver swords are one of the sort of classic examples of adaptive radiations. And so what an adaptive radiation means is that it's a group that speciates or radiates into various available ecological niches. And so we end up having this really wide variety of niches getting filled, and then we have these very different diversity of chute systems that vary in their growth form, their size, their shape, and their habit to be able to adapt to the resources that they need for growth and reproduction in those varied habitats. That's a cool kind of classic example of an adaptive radiation. So that's within a lineage, multiple different species, but of course we can actually have some variation in the chute system, just like we saw in the roots, in genetically identical individuals. And so remember that this is what we call plasticity. So plasticity where it's genetically identical. In different environments. And so these actually exhibit different morphologies in different environments, but they're genetically identical individuals. And so this is an experiment that was done by Sultan in 2010, looking at a plant called Persicaria, and looking at what happens when you vary moisture and light conditions in genetically identical individuals. And so down below, we have two genetically identical individuals here, which are grown in different environments. And so in those, so those different environments are on the right side, we actually have high moisture and low light. And on the left side, we've got the opposite of that. We've got low moisture and high light. And so these might mimic sort of two different types of environments that we would see. In the right, where we have high moisture and low light, this might be something like our forest environments, like our dark forests where a lot of light doesn't penetrate down to the soil, but moisture levels are high enough to have big trees around. Where on the left, we might have a situation where we see, like we see out in sort of south-facing exposures here, or out in the Columbia Basin, or out in the desert where we've got very low moisture content, but lots of radiation, right? Like a lot of high light environments. And when we do that, we actually see you can see just by looking at these two pictures, you can see pretty different morphological results in terms of when we have high moisture and low light, and low moisture and high light. And so I want to describe a few of the differences that we see here. So that plant on the left that was in those dry soils and full sun, we end up with more branching. So increased branching. Why might that be important? Well, we see this kind of seeking out, trying to spread out its crown, the branching structure in there. So we see more branching. The leaves are narrow. We have narrow leaves. And importantly, the leaves are thick. With a very thick cuticle to prevent that moisture, and thick mesophyll where the photosynthesis is happening. And that's both adaptations to water stress and preventing water loss. And then these have a high root biomass. And this, of course, is them searching for, investing in that below ground to search for water more efficiently, and reducing the biomass of the above ground, making these smaller, thicker leaves because it's not as hard to capture that light energy in this high light environment. On the right-hand side, where we see these grown in moist soils in a shaded environment, we have sort of the opposite. We have a less branched individual. We have wide, thin leaves with a thinner cuticle and thinner mesophyll or thinner photosynthetic tissue. And our biomass here is actually put into the leaves. So high biomass in leaves. And that's because light is the actual limiting factor here. We need to actually capture more light. So we have these thinner leaves that are bigger, wider, and spread out so that they are capturing more of the light in this. And so these are all responses that maximize photosynthetic surface area despite that lower biomass. Responses to maximize photosynthetic surface area. Okay. And so we see, just like we saw in the roots in these different environments, we have some plasticity in genetically identical individuals to be able to adapt to, or to be able to be suited to specific environmental conditions that each individual plant is experiencing. Plasticity is, this is a good opportunity to talk about how people study plasticity. Plasticity is something that's been studied for quite some time, and it's a really, some classic experiments have been set up to do that. And this this excerpt from your textbook sets up shows you the classic experiment of a group of researchers from the University of California named Clausen, Keck, and Heise. Clausen, Keck, and Heise. In 1945. And what the types of experiments that they pioneered are what are called common garden and reciprocal transplant experiments. And so this is a way to look at how much does a plant or whatever, an organism's growth form or their morphology, their phenotype, how much does the phenotype depend on the environment? And then, of course, sort of in the background of this is how much then depends on the genetics. And so this is recognizing that there are genetic components to phenotype and there are environmental components to phenotype. And this allows us to test the genetic versus environment axis in phenotype. So how much of that phenotype is due to the environment, and how much of that phenotype is actually due to the genetic background? And so in this, the question here is, how much does a plant's growth form depend on its environment? The hypothesis, we don't have necessarily an explicit hypothesis, but the goal of these are to, as I just said, explore that interaction between genetic and environmental influences on the plant, the overall phenotype of the plant and species in this case. And so the way that this experiment is set up is that cuttings are taken from individuals. This is a plant called Potentilla glandulosa that grows throughout the elevational range of the Sierra Nevada. So it grows at low elevations, medium, mid elevation, and high elevation habitats. And that's shown by the green, blue, and purple populations here, spanning a 3,000 meter kind of transect through California. Those individuals, they take cuttings from each of these and then propagate genetically identical individuals. And so they've got these propagules, or plants, growing from the high elevation, from the medium elevation, and from the low elevation. And they take these and then they grow these in common gardens. And so we're growing these all together in one place. We then take these cuttings from each of those and transplant them into each of the three environments. So we've got all three represented at each of these elevational sites where they came from. Sorry, I drew that last one wrong. Got one coming from here, one coming from here, one coming from here. And so we've got each one represented in each of those. This is the reciprocal transplant part of this. You're transplanting individuals from each source into each elevation habit, and you allow them to grow and then observe the results from there. And so by doing this, you can actually see what would our predictions be if we thought that that genetics were behind this. Well, if genetics were the only thing behind the differences in growth form in these three different places, then we would expect them to grow the same no matter where they are. If environment is part of it, then we would expect them to have differences in their home environment versus when they're reciprocally transplanted into different places. And so by tracking that, you can actually see what the results are, or what the result in terms of the environment. And so here's an example of mature plants that are observed. This is a high plant grown at low elevation. It has a small size. A high plant grown at medium elevation, it's got this sort of medium height, and then this high plant grown at high elevation, it's got this large size. The other thing that they're looking for in terms of size and biomass is the number of flowers, or the reproductive output, as a measure of fitness. And so what is the conclusion from this one? Well, it's that environmental conditions have an influence on body size and shape. So genetically identical plants, these are all plants that were from the high elevation. So these are all genetically identical. Genetically identical plants look different at each site. And so we end up with, although they're genetically identical, we end up with different phenotypes at each site. But that genetic makeup also has a large influence on their morphology. So it's not pictured here, but plants from each source population look different when they're grown in the same habitat. And so the common garden piece of this. So this is all the reciprocal transplant piece. So this is the reciprocal transplant result. The common garden result is this one here. So although when these plants are grown from the plants from each source population look different even when they're grown in the same habitat. And so this is the common garden piece of it. And so what's the take home from this? Well, there is phenotypic plasticity. So we do see that these grow differently depending on the environment that they're in. But we also see that there's a genetic component to that phenotype as well. So you've got both things going on. Okay, cool. So Klaus and Keck and Heisey kind of pioneered this. This is commonly used throughout not just plant systems, but animal systems as well, to look at phenotypic plasticity in phenotypic plasticity in different environments. Okay, just like before we did with the roots, let's look at some modifications to the chute systems and some of my favorite modifications to the chute system. The first of these shown here is a modification for water storage. And so here we see a cactus stem, and we see that it's enlarged. So an enlarged stem for water storage and photosynthesis. So why do I say and photosynthesis? Well, where are the leaves on this thing? If we notice, the outside, the stem itself is the green part, and that's where the photosynthesis is happening. These spines that are on here, these are actually the modified leaves. So leaves take on a different function in cacti, and that they are now a protective function rather than a photosynthetic organ. The photosynthetic organ itself is the stem in most cacti. So that's a cool modification to stem tissue, and a modified leaf there as well. There's a bonus. This is strawberries, shown on the next one. This is our strawberry example. And what I want to show here is a stolon. And stolons are these above ground horizontal stem. And the main function of this is actually asexual reproduction. If you've grown strawberries before, you've seen this. These stolons allow the plant to spread out horizontally along across the ground, and then each little plantlet here will actually root down into the ground, and then this stoloniferous connection, the stolon can break, and a new plant will form. And you'll see this spread throughout your strawberry patch in this way. Well, stolons are very common throughout plants, and so we see many different groups with stolons and for the same type of function. A similar type of organ or modification to stems is actually seen in these irises here below, the irises below, and that's the rhizome. And the rhizome is a below ground horizontally growing stem. So below ground horizontal stem. Similar function here in that asexual reproduction. And so just like with if you've grown irises before, you know that you can break off pieces of the rhizome and go and plant that elsewhere, and then a new plant will grow out from that. And so each place where there's each kind of node on here, we see roots forming and we see a new plant forming. And so asexual reproduction, another term for this asexual reproduction is vegetative reproduction, because it's reproduction, producing a new individual, but coming from a vegetative rather than a reproductive part of that. Rhizomes also have a secondary function that's important to mention. And so I'll just squeeze it in down here. Secondary function is actually starch storage. So iris is a good example to use for this. These die back every year, right? They die back down to the ground level every year, and they're storing those starches in that rhizome, and then they can generate a new individual, a new plant every year from that energy stored in that rhizome. One of my favorite examples of rhizomatous plant is actually what we see here in an aspen grove. So these are aspen groves, and this is a picture of an aspen grove in Colorado. But these grow everywhere. So this is Populus tremuloides, the quaking aspen. And so these are genetically identical. Identical clones, if you will, of each other that are connected through rhizomes underground. And so this modification basically, since we're talking about, records from the smallest, that duckweed, to the largest organism or individual in that giant sequoia, well, this could be argued to be the actual largest organism in terms of biomass, since these are all genetically identical clones from, basically the same individual. These will break their root connection, their connections, their stem connections, and separate themselves from the rest of the population. But this has been argued to be the actual largest organism in terms of biomass are these big aspen groves. But this is a good example of these genetically identical. And in this case, this is a great modification for a really common disturbance in these habitats, and that's fire. Right? Fire is a part of this ecosystem, and that's why we see these kind of even age stands in things like these aspen groves, because a fire or a disturbance comes through, wipes out everything, and then these will actually regenerate from below ground and create these stands of individuals again. Okay, a couple more modifications here to the chute system. This is the one, of course, that we're all familiar with, the tuber. And here we see these in potatoes. Other plants have tubers as well, but here's the potato. And what this is, a modified underground stem that's modified for starch storage. A lot of people think that potatoes are roots. They're not. They're actually a special type of underground stem. If you look at a potato, you can actually see a lot of the parts that we've talked about. So an eye of a potato, as we're used to calling it, well, those are actually nodes. And where leaves would have been attached, and we have internodes in between them. And the little eye part of that is actually an axillary bud. And so if you're familiar with how potatoes are actually propagated, you might know that you can actually just cut these up, and as long as you have an eye, an axillary bud associated with that chunk of potato, you can plant that in the ground and start a new individual from that. And that's the way that most potatoes are actually cultivated, is vegetatively through these modified underground stems or tubers. And of course, we exploit the starch storage there as an important food source. Okay, our last type of modified stem that I want to mention is a thorn. Thorns are modified stems for protection. And from herbivory, mostly. And so this on the right here is actually a picture of a bullhorn acacia. Bullhorn acacia is a plant that grows throughout the Central America and throughout Central America, and it's a really cool species. But these modified stems are for that protection from, like I said, from herbivory. If we look at, you know, how do we recognize this as a stem, we actually see, we can look to some of the other parts, and I'm going to just scroll up to a better example of this in this honey locust. And so these are honey locusts from, this is a honey locust that grows in the southeastern US. And what we see here is that the trunk of this is actually covered in these modified stems or thorns. And if you notice, you can see it down in this picture down on the right-hand side here, that this modified stem here, I'm going to actually zoom right in on that. This modified stem here, this is coming out, these leaves are coming out of the axle, this modified stem, or these thorns are coming out of the axle of where those leaves attach. And so that's actually positionally where we would expect a modified branch or a modified stem to come out of the axle of a leaf, because that comes from an axillary bud itself. And so we know that these are actually modified stems in this case, making them thorns. This particular tree I find totally amazing because of how intimidating it is. And so just to give you some scale of this picture, this is a trunk that's probably 18 inches or so in diameter from this honey locust tree. And the thorns on these can be 4, 5, 6 inches long. And so we get these thorns that are really long and branched thorns. And it's covering the whole trunk of this. So this is a really intimidating tree when you walk up to it with all of these thorns, this armament that's really on there. And so people have asked, like, why the heck does this plant in the southeastern US, so this is in the Appalachian Mountains, why the heck does this plant have these huge thorns all over it? And the prevailing hypothesis for this is something called the Gomphotherium hypothesis. Gomphotherium hypothesis. And this hypothesis relies on the idea that in the Pleistocene, we had, so upwards of 10 to 100 or 200,000 years ago, we had covering North America, we had a group of animals that we call the North American megafauna. And so this includes things like mammoths. These were tromping around North America eating plants. And this is, I'm sure, a very good protection from herbivory when you have these huge mammals that are breaking your branches off and mowing on them. And so this is the prevailing hypothesis for this. I think this is a cool hypothesis, and it's probably right on. Obviously one that we can't really test, but it makes a lot of sense given where these are growing and also sort of the age of these different lineages that have these big thorns on them, modifying the branches. So this is a really cool tree, and there's other things that have fruits that fit the Gomphotherium hypothesis. They're fruits that aren't dispersed at all today, but they likely were in the past dispersed by some of these megafauna that were covering North America at that time. One just added little side note about this one that I'll end this lecture with is that this honey locust, or Gladitzia is the scientific name for it. This is probably one of the most common street trees planted around in many places, but also around Moscow today. So downtown Moscow is actually lined with honey locusts. And you're probably thinking to yourself, well, I've never seen those thorns out there before. And you're right. It would be kind of crazy, although it would be really cool. It would be kind of crazy to have this as a street tree in downtown Moscow. But it is there. But what's grown, because it has really nice leaves. The leaves are all of these divided leaves like you see in the picture here. So they create a nice sort of open speckled shade sort of canopy. They're not a very messy tree in terms of the leaflets themselves are pretty small, and so they don't create a large mess. So they're a nice street tree, except for the fact that in the wild they've got these 6-inch thorns on them. But there are thornless varieties of them, and so Gladitia that are planted, the honey locusts that are planted in downtown Moscow are actually all thornless varieties. One of the cool things that you'll see right now in the springtime is that instead of having these clumps of thorns coming out of the trunk where those branches would be, you actually see when these plants start to put leaves on again in the spring, you'll actually see sort of groups of leaves or branches coming out of the trunk. So not clumps of thorns, but actually branches. And so this thornless variety that's been cultivated as a street tree does still have these branches coming out of the trunk where the thorns would be. But those branches are these leafy, nice, soft, leafy branches rather than these crazy big thorny branches. Okay, so next time you're in downtown Moscow, especially as the weather starts to get better and we start to see leaves emerging in the next couple of weeks, check out the street trees and notice that these branches are coming out. And then remember, what should be coming out are these 6-inch thorns, which would be way cooler for sure. All right, thanks. See you soon.